Good morning, everyone. Today I am going to take class on Bacillus. At the end of this class, you should know various species of Bacillus, the virulence factors and pathogenesis of each species, the clinical manifestations, how you will diagnose the organism in the laboratory, and the treatment and preventive measures. So far, you had classes on several bacteria. They were gram-positive bacteria, gram-negative bacteria also. So they were not spore-forming bacteria. So this class, Bacillus, they are spore-forming organisms. So the gram-positive spore-forming bacilli, they comes under two genera. One is Bacillus and the other one is Clostridium. So Bacillus, it is a obligate aerobe. That is, it will grow only in the presence of oxygen. And they produce non-bulging spores. Clostridium, they are obligate anaerobes. That is, they won't grow in the presence of oxygen. They won't tolerate oxygen. And they produce bulging spores. So this is the main difference between Bacillus and Clostridium. The same thing only. In the diagram, you can see here the Bacillus, they have non-bulging spores. The empty space you see is the spore, because it won't take up the gram stain. So the empty space here, it is a non-bulging spore for Bacillus, and Clostridium, it produces bulging spores. Bulging spores. Okay. So Bacillus, the genus Bacillus, it contains species which are pathogenic as well as non-pathogenic. The two species which are pathogenic are Bacillus anthracis and Bacillus cereus. Other members, they are found in the environment, in the soil, air, water, etc. They are also seen as contaminants in the culture media. Except the anthrax bacilli, all the other bacillus, they are motile with peritrichous flagella and they are non-capsulated. So anthrax bacilli, the Bacillus anthracis, it is non-motile, but it is capsulated. Coming to Bacillus anthracis. Bacillus anthracis produce a disease called anthrax, which is a zoonotic disease. So what is a zoonotic disease? It is an infectious disease in which the primary infection occurs in the animals and it can be naturally transmitted to humans. So Bacillus anthracis produce a zoonotic disease which is called anthrax. Another property of Bacillus anthracis is it can be used as a biological weapon. Okay. And on gram stain, you can see long rods arranged in chains. And there are non-bulging spores also. So this typical appearance is known as bamboo stick appearance in gram stain. And as I have said, Bacillus anthracis is non-motile, it is capsulated. Now the historical importance of this organism, Bacillus anthracis. Bacillus anthracis was the first pathogenic bacteria which was seen under the microscope and the first communicable disease transmitted by inoculation of infected blood. And Robert Koch first isolated this organism in pure culture. It was the first bacteria isolated in pure culture. And the Koch postulates were based on Bacillus anthracis only. And Louis Pasteur, he prepared the first live attenuated vaccine for anthrax. It was for anthrax that Louis Pasteur prepared the first live attenuated vaccine. Now, coming to the virulence factors and pathogenesis. The disease manifestation occurs because of the two main virulence factors. One is the anthrax toxin and the other one is the capsule. Anthrax toxin, it has three components. One is the edema factor, then protective factor and lethal factor. Of the three components, edema factor acts as the active fragment. The edema factor is responsible for the manifestations of the disease. This acts as a adenylyl cyclase and increase the host cell cyclic aim. As a result of this, there will be edema and other manifestations of the disease. Now, the second component, the protective factor, it acts as a binding fragment It helps in binding of the toxin to the host cell receptors, and it facilitates the entry of other two fragments into the host cell. The third component, the lethal factor, it causes cell death. This toxin synthesis, it is controlled by a plasmid. So if we can remove the plasmid, we can make the strain avirulent. The second virulence factor is the capsule. In many bacteria, the capsule will be made of polysaccharide. But here in anthrax, the capsule is a polypeptide capsule, which is made of polyglutamate. Capsule is also plasmid coated, and its mechanism of action, it is similar to that in other capsulated organisms. It inhibits the complement-mediated phagocytosis. Now coming to the clinical manifestations. Because it is a zoonotic disease, the primary infection occurs in animals. So the animal anthrax, it occurs more commonly in herbivores compared to carnivores. And this occurs because of the ingestion of spores present in the soil. When the animal ingests the spores, they get the disease. And spread from one animal to another animal is rare. The disease manifestation in animal is it will produce local cutaneous lesions, and fatal septicemia can also occur. And this infected animal, it will discharge lots of bacilli through their mouth, nose, and rectum into the soil. And these organisms, the bacilli that remain in the soil, which will sporulate, and they remain as source of infection for the human beings. Now coming to human anthrax. Human anthrax occurs mainly by through three modes of transmission. And rarely through the fomites also can spread. But the main modes of transmission are three, of which cutaneous mode is the most common. That is, the organism enters through the abraded skin, and this is most common in people who are occupationally exposed to the animals, infected animals. This will produce cutaneous anthrax. And the second mode of transmission is inhalational mode. This will produce pulmonary anthrax. This occurs when the spores are inhaled. This happens in people who are working in the wool factory. The spores which are present in the wool just get inhaled. The third mode of transmission is also rare. That is ingestion of carcasses of animals. If that happens, it will produce intestinal anthrax. So the three clinical types are cutaneous anthrax, pulmonary anthrax, and intestinal anthrax. Coming to cutaneous anthrax. This is also known as hide porters disease. This is because it occurs more commonly in the porters, the dog workers who are carrying hides and skin on their bare back. And the typical lesion that occurs in the skin, it is known as malignant pustule. Though it is named as malignant pustule, it is not a malignant condition. The term only is malignant pustule. Okay. So the disease begins as a papule, progress on to a vesicle, and finally, whole black colored necrotic eschar is found, which is typical of cutaneous anthrax. This is surrounded by non-pitting indurated edema. The cutaneous anthrax, it is a self-limiting condition only. Now the pulmonary anthrax, it is also known as wool sorters disease. This is because it occurs in those who are working in the wool factory. The spores present in the wool, when it is inhaled, the disease occurs. The manifestations include hemorrhagic pneumonia, and if the bacilli travels through the bloodstream, it will reach other organs and it produce meningitis, other infections also, septicemia, etc. And one thing is the pulmonary anthrax is a fatal disease if left untreated. Okay, so cutaneous anthrax is self-limiting only, it is not fatal, but pulmonary anthrax is fatal. And this is the most common mode of transmission which is seen in bioterrorism. Bioterrorism, you know, in 2001, United States, there was an outbreak and it is a highly fatal condition. And we can save the patient if we diagnose the condition early and antibiotics are started in the early stage itself. Now, the laboratory diagnosis of anthrax. This is an organism that poses high risk of laboratory acquired infection. So, utmost precautions should be taken while dealing the specimen. The specimen should be processed in appropriate biosafety cabinets with all precautions. And ideally, the specimen should be collected before the starting of antibiotics. The specimens to be taken are for cutaneous anthrax, that is a malignant pustule. You can take the pus as a swab or a pus aspirate. And in pulmonary anthrax, sputum is a sample. For septicemia, if we are suspecting septicemia, we can collect blood for culture. Then if there is meningitis, we can collect CSF. And for intestinal anthrax, gastric aspirate, feces or food can be used for culture. Then another sample is earlobes from dead animals. Now, direct demonstration, that is the first thing that you do with the specimen. It is the gram staining. So on gram staining, we can see gram-positive large rectangular rods. You should remember that spores are not seen in clinical samples. That is, when we receive a sample to the laboratory and we observe directly the gram staining, you won't be able to see the spores in the clinical samples. The spores are produced when we culture the organism in the laboratory and we take smear from the culture, we will be able to see the spores. Another reaction is McFadden reaction. That is, it is a staining only. We stain the smear with polychrome methylene blue, and we can see the capsule of the Bacillus anthracis. It is seen as a amorphous purple colored material surrounding the blue bacilli. We usually won't do this for human specimens. We use this for presumptive diagnosis of animal anthrax. Now, another important test is direct immunofluorescence test. This is used for the initial confirmation during bioterrorism. This will detect capsular and cell wall polypeptide antigens using fluorescent tagged monoclonal antibodies. Another important test that we do is Ascoli's thermoprecipitant test. This test is done when the sample is received in a very putrid form. So we assume that the bacilli won't be viable. So in that case, we won't be able to do the culture. No? So we will do Ascoli's thermoprecipitant test, which is a ring precipitation test. It is an antigen antibody reaction only. So what we do is we grind the tissue samples in saline and we boil it and take the filtrate. So this antigenic extract that we get, we take it in a narrow capillary tube, and over the antigenic extract in the capillary tube, we add anthrax antiserum. So there are two layers, one antigen and the other antibody. So there will be antigen antibody reaction happening at the junction. So there occurs a ring. A ring of precipitate is formed at the junction between the two in 5 minutes. So this is a ring precipitation test known as Ascoli's thermoprecipitant test. Now we do culture. So this is an aerobic organism, obligate aerobe, and it is a non-fastidious organism. So it will grow in ordinary culture media. And another property is it has a wide temperature range. It will grow between 12 to 45 degrees Celsius. And we can promote spore formation in culture at 25 to 30 degrees Celsius by using distilled water, 2% sodium chloride, oxalate, and oxygen. Now the colony morphology. On nutrient agar, you will see gray-white colonies which are irregular, round and opaque with a frosted glass appearance. Gray-white colonies which are round, irregular, opaque, and frosted glass appearance. So this is the frosted glass appearance of the colonies of Bacillus anthracis. And another feature that you see with the colony while observing under low power. You observe the edge of the colony under low power. You can see interlacing chains of bacilli, which appears as matted hair, locks of matted hair. So it is termed as medusa head appearance. This is the medusa head appearance of the colonies of Bacillus anthracis, observing under low power of the microscope. Now, coming to the appearance in blood agar. On blood agar, the colony will be dry, wrinkled, and non-hemolytic. You should remember that on blood agar, Bacillus anthracis will appear as non-hemolytic, while other species of Bacillus, they will appear as hemolytic. They are known as anthracoid bacilli. Bacilli which are not anthrax bacillus, that is the species other than the anthrax bacillus. They are known as anthracoid bacilli, and this anthracoid bacilli will be hemolytic, while the anthrax bacilli, Bacillus anthracis, non-hemolytic. Both of them will give only dry wrinkled colonies on blood agar. And another pattern that you see in the gelatin stab agar. In the gelatin stab agar, what we are observing is the gelatin liquefaction. Okay, so we are observing it in a test tube. In the test tube, the gelatin agar is there, and we inoculate the organism onto it, and this will liquefy the gelatin. The organism will liquefy the gelatin. This liquefaction is maximally seen at the top, and it will decrease while going down. So this will give an inverted fir tree pattern on gelatin stab agar. And you should remember the selective media, they are solid medium with penicillin and pledge medium. And on solid medium with penicillin, what happens is by the action of penicillin, the cells will become, cell wall will become weak. So the cells will enlarge, and they appear in chains also. So this will give a string of pearl appearance in solid medium with penicillin. So it is a selective medium for Bacillus anthracis. Another selective medium is the pledge medium, which contain polymyxin, lysozyme, EDTA, and thalassacetate in heart infusion agar. So these two are the selective medium that we use for Bacillus anthracis. So this is a picture of gelatin liquefaction. Here you can see maximum liquefaction happens at the surface and it decreases down. So this will give a inverted fir tree pattern, okay, of gelatin liquefaction. Now coming to the culture smear. When we take smear from the culture, you can see spores also. So the spores are seen as empty spaces. Typical appearance is bamboo stick appearance. And you can demonstrate the spores by using special stains. So the spore won't take up the gram stain. So you can demonstrate it in special stains by using malachite green, or the spores are acid fast also. Bacterial spores are acid fast. So you should use 0.25% sulfuric acid as the decolorizer for acid fast staining in order to observe the spores. Now, lipid granules, they take up the Sudan black pea film. Now, the serological methods for diagnosis, you can see antibodies. The antibodies, they appear in convalescent sera, and this can be detected by ELISA or immunodiffusion in gel. And a molecular diagnosis, it is the confirmatory method for diagnosis. And this is done by using specific primers, PCR with specific primers. And for epidemiological purpose, we can do a molecular typing also in order to trace the source of infection. The CDC has given some guidelines for diagnosis of anthrax at the time of bioterrorism attacks. So they have given, like, for presumptive diagnosis during the bioterrorism attack, if you are seeing any large gram-positive bacilli with a morphology and cultural features similar to anthrax, you can presumptively tell that it may be Bacillus anthracis. And we have to confirm now For initial confirmation, we do the direct immunofluorescence test or lysis by gamma phage, and further confirmation, final confirmation is done by PCR. So these are the guidelines given by CDC for diagnosis during bioterrorism attacks. Now coming to the treatment and post-exposure prophylaxis for anthrax. For treatment, we give ciprofloxacin or doxycycline, and we add clindamycin with or without rifampicin. So this is the treatment. You should give ciprofloxacin or doxycycline plus clindamycin with or without rifampicin. This should be given for 60 days. Okay, and post-exposure prophylaxis also we have to give ciprofloxacin plus doxycycline or amoxicillin we can give. If the strain is penicillin sensitive, the organism is penicillin sensitive, we can give amoxicillin, otherwise give doxycycline. This also should be given for 60 days. And there is one more treatment option. It is a monoclonal antibody which is directed against the protective antigen. This can be given for inhalational anthrax. The name of the antibody, monoclonal antibody, is raxibacumab. This is another treatment option for anthrax. Now the preventive measures. The general preventive measures that you follow is that you should follow to prevent the occurrence of the diseases. The disposal of animal carcasses by burning or deep burial. Then decontamination of the animal products by autoclaving. Then you should wear protective clothing and gloves. Those who are occupationally exposed to animals should follow these general preventive measures. And there are two vaccines. One is for animals. It is called Stern vaccine. It is a live attenuated, non-capsulated vaccine. It is a spore vaccine. The component that we use for production of vaccine is the spore of the organism. This is a spore vaccine. So this is not given for humans. It is given for animals. It gives 1 year protection. The second vaccine is a toxoid vaccine. It is a toxoid directed against the protective antigen. Toxoid prepared from the protective antigen. Okay. It is an alum precipitated toxoid vaccine. It can be used for human beings in occupational exposure, during occupational exposure. The 3 doses have to be given. It is given intramuscularly at 0, 6 weeks, and 6 months. And a booster dose is given after 1 year. So these are the preventive measures that we can take to prevent the occurrence of the disease. Now the anthracoid bacilli, the Bacillus species other than the anthrax bacilli, they are termed as anthracoid bacilli. Of the anthracoid bacilli, all are non-pathogenic except the Bacillus cereus. Okay, so they resemble anthrax bacilli. They have dry wrinkled colonies, and on gram stain there will be chains of gram-positive bacilli, etc. They are actually seen as a laboratory contaminant, but they are not anthrax bacilli. And the main difference I told earlier, they will be hemolytic, while the anthrax bacillus is non-hemolytic or blood agar. There are several other differences also. You can go through that in the textbooks. Now coming to Bacillus cereus. Bacillus cereus is normally present in the soil, and it is isolated from food like milk, meat, poultry, etc. And it is known to cause food poisoning. Bacillus cereus will cause food poisoning. Other than food poisoning, it can also cause other infections like ocular disease, keratitis, ophthalmitis, etc., panophthalmitis, etc. after trauma. And it can also cause a spectrum of illness like endocarditis, meningitis, osteomyelitis, etc. in presence of medical devices. So the main thing that you should remember is Bacillus cereus, it produces food poisoning. It is known to cause food poisoning. And there are 2 types of toxin. One is a diarrheal toxin, and the second one is the emetic toxin. This diarrheal toxin, it produces mainly diarrhea. Emetic toxin, it will produce vomiting and abdominal pains like that. So this is a main difference between the diarrheal type of food poisoning and the emetic type of food poisoning. So for diarrheal type, the incubation period, that is the time of occurrence of the symptoms after taking the food. that is 8 to 16 hours. It is longer compared to that emetic type, 1 to 5 hours for emetic type. Why? This is because of the preformed toxin that is formed in the food itself. In emetic type, the toxin is present in the food itself, the food that we take. So the illness will manifest earlier. The incubation period is 1 to 5 hours only. But for diarrheal type, the toxin, it is not present in the food, but it is produced in the intestine. It is secreted in the intestine. So it will take more time. The emetic type toxin, it is heat stable, while the diarrheal type is heat labile. And the food items that are implicated are Chinese fried rice is implicated in the emetic type, and meat, vegetables, cereals, etc. They are implicated in a diarrheal type. From other food items also we can get, but these are the food items which are most commonly implicated. And the clinical features for diarrheal type: diarrhea, fever, abdominal cramps, and emetic type, vomiting and abdominal cramps. Now coming to the laboratory diagnosis and treatment of Bacillus cereus. The specimen that you collect for laboratory diagnosis is the feces, and we should inoculate it into selective media for isolation. The selective media are mannitol, egg yolk, polymyxin, phenol red agar, MIPA, and PEMBA, polymyxin B, egg yolk, mannitol, bromothymol blue, agar. So these two are the selective media for Bacillus cereus, MIPA and PEMBA. And it is motile, it is non-capsulated, and usually it is sensitive to clindamycin, erythromycin, vancomycin, aminoglycoside, tetracycline. So these are the treatment options, and will be resistant to penicillin and trimethopril normally. That is about Bacillus cereus, and there are some Bacillus species which have other uses also. Examples are Bacillus thuringiensis. It is used as a larvicidal agent for mosquito control. Then Bacillus styrothermophilus, Bacillus subtilis, Bacillus pumilis, and Blobici. These are all used as sterilization controls in various methods of sterilization. Bacillus styrothermophilus we use in autoclave, and Bacillus subtilis we use in plasma sterilization. Bacillus pumilis in ionizing radiation, and Bacillus blobici in ethylene oxide sterilization. So these are the other Bacillus which can be used as a sterilization controls, and also other uses are there. As a larvicidal agent, Bacillus thuringiensis is used. So that is all about Bacillus. Thank you.